Well, we're back and we're getting ready to talk a little bit more about linkage. Now, in the last video, we talked about linkage mapping and really leading up to the point where Alfred Sturtevant was the one who constructed the first genetic map of Drosophila. So remember, this is several years after Thomas Hunt Morgan identified sex linkage in Drosophila. And of course, once he had that tool, he had a very, very smart undergraduate student, Sturtevant, of course. And I think it's critical, or at least interesting, and maybe should motivate you guys a little bit, that Sturtevant was an undergraduate of Hunt Morgan, and he was the one who actually started off this concept in the mathematical background between to identify linkage amongst genes. Now remember, it wasn't until the 60s that we actually had a single gene mapped to an autosome in humans, okay? Now just a little reprise, because I think it'd be interesting for you guys to see what actually Sturtevant did. Notice he dealt predominantly with the X-linked chromosome. Now remember, he talked about the X chromosome in Drosophila, of course. The X chromosome is chromosome 1, and took triple homozygous mutants. Okay, so take a peek at those triple homozygous females that are over here. They are homozygous for scoot, okay, echinus, which have no cross veins in the wings, and of course cross veinless. Now, they were crossed with a wild type male, and remember, look over here, all the wild types in Drosophila, okay, have the little plus sign. So in Drosophila, the genes are named after the phenotypic effect of the gene. The mutant is just the letters, and the plus sign indicates the wild type allele of that gene. So by crossing a wild type male to a completely homozygous female, end up with females that are completely heterozygous. So then, by crossing them with a triple homozygous male, they are able to create individuals that are always able to identify linkage and recombination. And they can use the phenotypic categorization of these individuals to calculate recombination frequencies. The data are listed below for you. And here are the basic ideas of the calculation. I think it's really fun to be able to look at, and I know my definition of fun is a little different than perhaps yours, is looking in these females, okay, and looking at single and double crossing over events and actually being able to calculate the recombination frequency. Remember from the last lecture that recombination frequency is basically equal to the map distance, and we call that map distance a centimorgan after Thomas Morgan. And so we see here, these are our references to centimorgans again. So we've got this great tool to be able to calculate recombination distance on the X chromosome, but remember, it's only chromosome 1 in Drosophila. And here is what Sturtevant was able to put together at the time, again in 1913, showing a genetic map, just as we did. But remember, here he's got 7 different mutations and 7 different genes, and the map distances among and between them. Heck of an undergraduate research project, don't you think? And I really enjoy thinking about when I was an undergraduate. I got to tell you, I think this was a little beyond me as well, but really something to aim for, something that's possible even without tools biochemical, molecular biology, all these tools that are available to us these days. It's really just a brilliant genetic model and some great minds to design experiments. That's what science is really about, and one of the reasons I like to present these slides. If we take a peek at what was done after Sturtevant, we see that this is a contemporary genetic linkage map of Drosophila. If you take a peek at all of the different chromosomes, here of course is chromosome 1 or the X chromosome, then we've got 2, 3, and notice how small chromosome 4 is, and only a very, very small part of chromosome 4 actually undergoes recombination. It is so small, chromosome 4 generally is thought not to undergo any recombination events. So we see, if you take a peek down the list, you can find all sorts of different mutations, and these are all phenotypic mutations. So again, if we're actually going to do this in the laboratory by breeding Drosophila, think of how many triple mutants had to be used in order to calculate the recombination frequencies of all of these different mutations. And so if you take a peek, I'm sure you'll be able to see, because I'll point it out to you, there's brown eyes. That was from our laboratory experiment looking at brown and scarlet. And of course, you can find scarlet as well over here. So you can see, because brown is located on chromosome 2, scarlet eye is on chromosome 3, that's why our total expectation of the F2 dihybrid between these mutants is 9 to 3 to 3 to 1, because they are in fact on different chromosomes. Now, again, I want you to think about the complexity of having to make all these crosses, count all these flies, and create this. And this is without any of the molecular biology tools we will be talking about. But you'll see how they fit together shortly. I like to take a brief divergence, because no genes were mapped to autosomes in humans. So the work in Drosophila, the work in plants, even the work in chickens, if you can imagine, was progressing at a pretty rapid rate throughout the 1910s through the 1930s. And a lot of genes were mapped, and very, very simple linkage maps were created. However, nothing in humans. Why? Obviously, we can't create our own sets of triple mutants in humans and dictate our own crosses, okay? So it really took a unique observation by a guy named Roger Donahue. And Roger Donahue in 1968, I'm sorry, we'll back up just a sec. So here's a classic way that we knew that some genes were on the X chromosome in human beings. And really, this is Queen Victoria's pedigree. And if you take a peek, here's Queen Victoria. Queen Victoria was the source of all the different hemophilias throughout the royal families in Europe. Here's the great pedigree showing that it has sex-linked inheritance. And it wasn't until, again, think about this for a moment, 1905, the human sex chromosome was discovered by Nettie Stevens and Edmund Wilson. Curious to me is it's one of the few times in science that Stevens actually got credit. And being a woman, you would think that Edmund Wilson would have nudged her out, but no, Nettie Stevens is credited as having identified the X chromosome. So kind of nice. So the Duffy blood groups were identified in the 50s. And you remember we talked about the different blood groups, the obvious ABO, the L and M blood groups identified by Landauer, and of course the rhesus factor, Rh plus and Rh negative. So these are the Duffy blood groups. They were identified in the 50s, and they were named for the Duffys from the patient with hemophilia who received multiple blood transfusions. And this was the way they discovered the antigens. At the time, only Duffy A and Duffy B were identified, but years later, 3, 4, 5, and 6, and notice they changed the nomenclature. This is very, very common in genetics, sadly, where they start labeling them A and B, and then they start finding more proteins, and they start identifying them with 3, 4, 5, and 6. This is done very, very commonly with, for example, the zonapalooza series of proteins, which were originally 1, 2, 3, and then A, B, C, and now they're back to 1, 2, 3. So science nomenclature is sometimes a little bit difficult to follow. And Roger Donahue is an interesting guy. Again, an undergraduate. He was taking genetics class at Harvard. And while he was in class, they did a couple of typical genetics experiments in laboratory. One was looking at blood types and creating a pedigree amongst your family. So they had little blood typing kits, and they would give the students enough to take home and actually do a pedigree of all their family members. We don't do that anymore. But they would also do karyotyping, and they would get them to actually put together a karyotype of their own genome. So again, take a blood sample, isolate the white blood cells, arrest them in metaphase, plate them, put them under a scope, stain them, and actually put together the karyotypes. Now, Donahue noticed, in fact, I sometimes wonder what one might have done if here is what I'll call the wild type. That's your typical set of X chromosomes. And of course, Duffy noticed that his X chromosome, one of them, was longer, and this area in here was very much lighter than everybody else's. So think about what you might have done as an instructor if a student kept coming to you and saying, oh yeah, look at this, I don't get it. You'd probably send them back to do it again to the scope and have them draw more blood, isolate the cells, etc. As it turns out, this is known as a heteromorphism. A heteromorphism, different shape. It is a heteromorphism because he has a differently shaped X chromosome. Duffy, however, or rather Donahue, however, was not willing to let this observation pass. And what he did was he actually went back home, and in addition to doing a blood test for the Duffy A and B groups, he also identified individuals in his family that did and did not have these elongated X chromosomes. And so he put together a pedigree, and he called the heteromorphism coilers and uncoilers, assuming that the DNA is less dense in that lightly stained area, and thought, well, this uncoiler element could be the basis of a mutation in the DNA, or perhaps an inversion. That is, the DNA flipped over during meiosis, and so as a result was a different shape than all of the other X chromosomes. And this was really the beginning of linkage analysis. And you can kind of take a peek over here. You see there is Roger Donahue, okay? And he indeed had the X chromosome. It's filled in with the marker. Anything with this white and black area, these individuals that have the black have the marker. Anything that's clear is basically wild type, a typical X chromosome, and you can see the inheritance, and it is associated, of course, with the Duffy blood group. So really an exciting observation in the 1960s by another undergraduate. So the entire area of linkage analysis is really due to two undergraduates. So we would not have our typical genome and human genomics and our genomic maps if it were not for these two individuals, Sturtevant and Donahue. Later on, I thought it'd be nice just to show you that what actually happened to the chromosomes. Here, of course, on the right-hand side is the normal chromosome, and on the left-hand side is the Duffy. And what you can actually see here is that it is this area of the chromosome that became inverted, and as a result, in this particular area, the chromosomal DNA did not supercoil properly, and so it became elongated. And if you kind of notice, what this really means is and I'll back up, when we look at this uncoiler element, notice that he's got it oriented, that the long area is here and the short is here. They are actually upside down from the actual orientation of the chromosome. So he actually had it upside down. Again, there's no way to tell top and bottom on a linear chromosome. Now, the fun part is if we take a peek now at chromosome 4, this is just a typical chromosome. I just chose arbitrarily chromosome 4 because there are a large number of disease genes located on chromosome 4. Some of these have been identified in and of themselves because of pedigree analysis. However, in fact, most of these are identified not by pedigree analysis, but by using molecular and biochemical genetics to identify these particular DNA sequences. So that will really constitute the next area of conversation for us, and we'll be able to talk about exactly what kinds of molecular and biochemical genetics are being used to create genomic maps. Hope you enjoyed that. It's a fun topic for me. Next up, more fun stuff.